Well then, um, good afternoon again. I'm Jeffrey Bennett. I'd like to thank the organizers for inviting me to talk in this session. And what I would like to do is first present my disclosures here, and then begin by reminding everyone that there are 19 approved therapies for the prevention of disease activity in multiple sclerosis. And in 2017, the era of targeted B-cell therapies officially began with the introduction of Ocrevus into the therapeutic landscape. And while other highly efficacious therapies were already on the market, anti-CD20 therapy provided new benchmarks for safety and efficacy and renewed a long-standing interest in a suspect in MS pathophysiology, and that is the B lymphocyte. My talk today is designed to tell you how to manage MS. Time is too short for that here, and there are too many variables associated with treatment internationally. Instead, I want to take the opportunity to focus on the landscape of MS therapeutics through a B-cell lens, and that is to review the evidence supporting B-cell involvement in MS pathophysiology and examine how it might inform us about the mechanisms driving success of current therapeutics and assist us in optimizing their use for further treatments. There's always been an abundant evidence supporting a role for B cells driving MS disease activity. Type 2 MS lesions are the most common observed in tissue pathology, and there are ectopic B cell follicles overlying cortical demyelinating lesions. IgG and complement deposition are observed in active lesions, and you can see IgG deposition and evident in this case of acute optic neuritis on histopathology, as well as in acute MS plaque. In the CSF of MS patients, there is clonal expansion of B and plasma cells, accompanied by persistent oligoclonal banding, a pathognomonic feature of multiple sclerosis, but not ideally specific. And importantly, both elevated B cells and an elevated IgG index have correlated with disease progression. In 2017, the first B-cell therapy, Ocrelizumab, was approved into the market, and recently Ofatumumab has joined it. They both have shown a robust ability to diminish relapse activity and new MRI lesion formation, disability progression, as well as the rate of brain atrophy, and you can view that list here. They reduce biologic markers of neuronal injury, such as serum neurofilament light, and they have shown some level to improve disability in treated patients. Clinical efficacy, although, has been somewhat less impressive, but noteworthy in the treatment of progressive MS, which Ocrelizumab has been officially approved in the US. Importantly, treatment with either agent was well tolerated, and there's no significant infectious signature in the short term. However, long-term treatment has been associated with about 20% of patients achieving hypogammaglobulinemia, some with infectious signatures. So, if you're going to treat with anti-CD20 therapies, is there an obvious difference? Well, on the surface, Ofatumumab given subcutaneously monthly, Ocrelizumab IV every 6 months. But it's important to note that they have different effector functions for antibody depletion of CD20, antibody-dependent complement, antibody-dependent cell-mediated cytotoxicity for Ocrelizumab, and complement-dependent cytotoxicity for Ofatumumab. This difference may impact the depth and extent of B-cell depletion. As noted here, both antibodies look like they fully suppress anti-CD20 presence in the serum. However, the noise down in this lower region is not really evident on this scale. And interestingly, in a murine model that has humanized CD20, labeled Ocrelizumab and Ofatumumab showed different penetration into axial and inguinal lymph nodes, with subcutaneous route of administration showing more penetration, and subcutaneous Ofatumumab impacting more penetration than IV Ocrelizumab. With that being said, it's interesting to note that Ocrelizumab has shown a difference in antibody exposure based on weight, with antibody exposure being correlated with lower disability expression, that is higher exposure, less disability expression. A similar notion, that is a dependence on weight has not been observed in the primary trial reports from ofatumumab. Any relationship between B cell count and disability progression, however, has not been presented. So how might we translate these B cell therapies into a view of how current MS therapeutics are acting and how we may behave and develop drugs for the future? IgG secretion and antigen presentation, pro-inflammatory and regulatory cytokine secretions have all been modes of actions of B cells that have been implicated in MS disease activity. Let's take a look at them individually. Antibodies have showed histopathologic and CSF evidence for involvement in MS disease. I've already mentioned those previously. Interestingly, we have cloned MS antibodies from the cerebrospinal fluid of patients and showed in ex vivo cerebellar slice cultures that the administration of these antibodies with human complement shows significant demyelinative activity as opposed to an isotype control. If we inject into the cerebral cortex of mice, similarly, we find large lesions with MS recombinant antibody, but no lesions with isotype controls. What seems to be in contrast is that antibody levels do not correlate with anti-CD20 therapeutic outcomes. What I'll bring to your attention, however, is this may not be necessarily a clinically meaningful metric for what antibodies contribute. That is, that it may not be that they are very impactful on clinical relapse or MRI lesion formation, and similar results are shown in neuromyelitis optica, where there's a lack of correlation with an overtly pathogenic aquaporin 4 IgG autoantibody. So if we look a little bit more closely, we might see some other effects that we should pay attention to. For S1P inhibitor Fingolimod, it does not reduce peripheral B cell blood concentrations, and in fact, it increases CSF naive B cells and decreases CSF plasmablasts without affecting overall CSF numbers. However, when we look at clonal populations and B cell repertoires, we find that after Fingolimod treatment, there is a reduction in the amount of clonal populations after 6 months of therapy. While the overlap of clones is similar, when we observe the persistence of clones in the CSF of the same patient, we find that Fingolimod contracts the CSF clonal population, and the only clones present are ones overlapping from an intact blood-brain barrier transit of these cells and not from a persistence of clonal populations within the CSF, indicating that there may be a CSF penetration and impact on germinal center activity. If we look at antigen presentation, B cells are potent antigen presenting cells that are dependent on their B cell receptor on the cell surface and the presence of co-stimulatory molecules such as major histopathcompatibility complex. Reduced antigen presentation has been noted in B cell treated patients, with an interesting facet that repopulating cells have enhanced antigen presenting activity and enhanced expression of co-activating molecules such as CD25, CD69, and CD40. In fact, B cells repopulating following rituximab treatment have been shown to induce CD4 and CD8 proliferation. Looking at cytokine expression in immune regulation, B cells participate with other adaptive and innate immune populations in a vast cytokine network that drives local and global pro-inflammatory and regulatory networks. B cell depletion decreases pro-inflammatory signaling, and this can be seen with changes in the ratio of CD4 to CD8 cells, as well as a decrease in the expression of pro-inflammatory CD4 and CD8 populations. B cells after repletion show reduced pro-inflammatory expression with reduced GM-CSF and enhanced IL-10 anti-inflammatory cytokine expression. And this can be shown in the graph on the right, where IL-6 presentation, or at least the fraction of IL-6, doesn't seem to change. Interestingly, the co-incubation of these B cells that are reconstituting induce myeloid cells to have a less inflammatory phenotype, with lower IL-6 secretion and higher IL-10 secretion. So how do some of our current high efficacy therapies impact these populations in similar or discrete ways? Cladribine and Alemtuzumab are impactful on CD memory B cell populations. Both of these agents impact the level of CD19 memory B cells in the long term, and oral and subcutaneous Cladribine causes a prolonged depletion of memory B cells, both class switched and non-class switched B cells. A similar effect is observed with Alemtuzumab. However, there's a significantly higher incidence of early high-grade lymphopenia with the use of Alemtuzumab compared to Cladribine. It's interesting that both resulted in a significant reduction in CD25 positive memory B cells, which may indicate a secondary impact, as we noted before, on antigen presentation activity. In a new adopted transfer model in the mouse with enhancement of IL-17 producing T cells that are resident in the meninges, it was noted that Siponimod also reduced Th17 T cells in the meninges, coincident with a reduction of B cells present in those meningeal-like follicle areas. And that could be interesting, as noted about with regard to what I previously mentioned with B cell repertoires. Dimethyl fumarate and monomethyl fumarate modulate B cells and cytokine secretions in another anti-inflammatory manner. Both of these agents reduce memory and mature B cells, and the B cell numbers that are present show an increased ratio of naive to memory and translational to mature B cells, as well as decreased pro-inflammatory B cells expressing GM-CSF, TNF-alpha, and IL-6. When stimulated, B cells express lower amounts of GM-CSF and lower amounts of TNF-alpha, possibly favoring a little bit, as shown here on the right, dimethyl versus monomethyl fumarate. If we look at the final arm of B cell regulatory signaling, we see that regulatory B cells, those that are presumed expressing IL-10, have a significant suppressive effect in various animal models of autoimmune disease. IL-35 regulatory B cells, LAG-3 positive regulatory plasma cells, and gut-derived IgA plasma cells have been shown to have a natural induced regulatory post-germinal center genesis and effect. And these populations may be modulated by MS therapeutics. On the surface value, it might think that it might be a little bit counterproductive to have B cell depletion of potentially regulatory cell populations, but it should be noted that anti-CD20 therapies will not directly affect already generated plasma cell populations due to the lack of that surface marker. And it could be that the modulation of other B cell populations and the lack of generation of pro-inflammatory populations that need to be repleted may alter the balance between pro-inflammatory and regulatory plasma cell populations. Indeed, in a recent poster presentation, again using this IL-17 murine model of meningeal inflammation and CNS inflammation, anti-CD20 therapies promoted the population of IgA plasma cells in the CNS meninges, possibly associated with enhanced BAFF-A production. But outside of B cell depletion, other therapies may impact this regulatory B cell population, and an illustrative example is Alemtuzumab. We all think of Alemtuzumab reducing pro-inflammatory T cell populations, and I already pointed out that Alemtuzumab has a profound effect on pro-inflammatory B memory cell populations. But here is a case report of Alemtuzumab treatment on a patient with 2 courses of Alemtuzumab experiencing 2 sequential relapses several months after their first and second cycle of infusion. These relapses were not associated with a significant increase in the memory B cell population as shown here and here. While this memory B cell population was a little bit expanded, it was significantly reduced from the prior monthly cycle. But what was more interesting is that while initial treatment caused a rise in regulatory B cell expression, each relapse was associated with a significant reduction in the presence of regulatory B cells. In fact, in a follow-up study looking at populations treated with alemtuzumab and using a surrogate surface marker of regulatory B cells, that is CD24 high, CD38 high B cells, alemtuzumab treated patients in remission had a higher mean number of regulatory B cells than their relapsing counterparts. And this could be a potential clue not only to the action of alemtuzumab, but the action of other MS therapeutics. What is a caveat, however, is CD24, CD38 B cells are not necessarily regulatory, as many repopulating transitional B cells follow under this same surface marker expression. So how do we look for the future? Is it important to keep on depleting, or do we look at other ways of manipulating B cell populations that could impact these same therapeutic endpoints? And in this case, I'll bring up one of the current MS therapeutics under development, that is Bruton's tyrosine kinase inhibitors, or BTKI. BTK inhibits the development of B cells as well as the activation of B cells at certain set points during their maturation cycle, as shown here. And BTK is expressed not only in B cells, but in dendritic cells, mast cells, and myeloid cells, including microglia within the central nervous system. BTK inhibitors in phase 2 trials have already showed some efficacy on the formation of new gadolinium-enhancing lesions. Unfortunately, therapeutic benefits will require the subsequent performance of phase 3 trials that are currently underway. But what's exciting about BTK inhibitors is not only their ability to modulate these B cell populations, but also to impact within the central nervous system other innate immunities. And taking a look more closely at that, from a mouse model, it has already been shown that BTK inhibition can block B cell maturation and decrease activation markers not only on B and T cells, but also decrease activation markers and function on myeloid cells. More importantly, it does this without the active depletion of these B cell populations. In antigen presentation assays, B cell antigen presentation is inhibited and B cell cytokine production is inhibited. However, both apparently on pro-inflammatory interferon gamma, IL-6, as well as anti-inflammatory arms. Instead of using this mouse model, we have used a model of cortical MS lesion formation that we have developed in collaboration with Ethan Hughes and Wendy Macklin. In this model, we apply MS recombinant antibody cloned from patients with human complement on the surface of mouse brains and use 2-photon microscopy to watch inflammatory activation. And recently, Helena Barr from Ethan Hughes' lab has presented her findings at the World Congress and the recent ACTRIMS meeting, showing that this antibody and human complement application shows profound microglia activation, as shown in red. And these microglia come and engulf in this montage over 24 hours, myelin enwrapping axonal sheets, and the systemic application of BTKI inhibits this reaction. To sum up, we've entered a new era of MS therapeutics led by the introduction of highly effective B cell depleting agents. There is ample evidence to support an integral role for B cells in MS immune pathogenesis. Experimental and animal models indicate that anti-CD20 and other MS therapeutics result in significant changes in B cell populations, both outside and potentially inside the central nervous system. Future studies are going to be needed to link these effects to meaningful clinical and immunologic outcomes in affected patients, to proactively prescribe, modify, and sequence therapies for the care of our MS patients. And before I end, I'd like to thank the organizers once again, as well as the people in my lab, Greg Owens, Kristen Schaller, Alana Ritchie, Yi-Ting Liu, of course, our collaborators, Wendy Macklin, Ethan Hughes, Katie Givens at the University of Colorado, and Marcus Kowarek, whose data in my lab we presented now at the University of Tübingen. Thank you very much for your attention.
Thank you, Jeffrey. It was excellent, a very comprehensive talk. So it's virtual applause. Thank you very much. I can see there is one question. Could the depletion of B cells be involved with downregulation of inflammasome signaling at oligodendrocytes and neurons throughout MS? Could be this pathway the key target of B cell depletion? If you open the question, yeah.
Okay, thank you. Well, I think that certainly B cells have a strong history of inflammasome secretion, as well as there has now been noted to be pro-inflammatory expression by astrocytes as well as glia within the nervous system in discrete single cell populations. And so therefore, I think that this is an important avenue to be investigated as well amongst the many features that I looked at, but we're going to have the appropriate models. And as we look forward, right now we've been using an excellent model of, I would argue, MOG IgG disease to study multiple sclerosis, a CD4-based disease. We need some more models, and I'm hoping some of the ones I mentioned previously with ex vivo amplification of pro-inflammatory T cell cytokines that populate meninges and reproduce some of the histopathology of MS. The antibody-mediated models we've been producing can allow us to purify these inflammasomes and analyze both their content and effect on this glial cell network that's going to be so important. But I think it's a good point and a primary target that we can look at.
Excellent, very interesting. And I wanted to ask you, we have now so many anti-CD20 therapies and more are coming, and then we'll have the BTKI to complicate choices. So how do you think this is going to go? Do you think that every new treatment is going to be used instead of the previous one, or there is going to be a niche for the use of all the anti-CD20? So yeah, how do you think this is going to evolve?
I think new anti-CD20s are going to, have a similar impact clinically to one another. What will be very interesting is as they get introduced, as we've already seen, with different methods of administration, the convenience for our patients will become an issue. The impact on vaccination schedules will become an issue, as we've already seen in the COVID crisis. The discontinuation of therapy in order to get anti-CD20 antibodies out of the system to allow for a better vaccine response will become important strategies, as well as we're going to have to become proactive for vaccination for patients before we treat with anti-CD20 agents. Overall, we're going to have to meld that together with the idea that maybe, as I pointed out, penetration and depletion extents may be quite different based on the action of the antibody, its penetration into the tissues, and that may have important weight-based effects that may require different dosing in different patients or no concern for dosing in certain patients. And overall, I think the efficacy and the safety we've observed so far, knock on wood, that long-term issues don't become more critical, but our experience with anti-CD20s sort of would argue against anything totally surprising would be, I think it becomes a benchmark for comparison. And what we have to optimize is whether depleting everybody or a focused target of understanding this better, as I tried to present, would allow us to say, can we acquire this with other drugs we're currently on? And when you fail another drug, Is there something that depleting all B cells is going to bring to the table, or adding other molecules may help to modulate the gap that you're missing?
Yes, very sensible, very interesting. Let's see an update next year. Thank you very much.
Yeah.
Thank you very much.
All right. Thank you.
Thank you. Take care.